It's my pleasure to welcome to the podium Professor Mike Sadowsky. Mike is a fellow in the prestigious American Academy of Microbiology, is internationally known and respected for his research work in the area of environmental microbiology. He currently is a co-director of the Microbial Plant Genomics Institute. He has published more than 100 original articles, and his work is widely cited by researchers in several scientific disciplines. A professor at the university's Department of Soil, Water, and Climate and the Biotechnology Institute, Sadowsky has been studying the symbiotic relationship between leguminous plants and rhizobial soil bacteria. He has focused on a process called nodulation, by which bacteria from root nodules help fix nitrogen in these plants, allowing them to thrive and reproduce in nutrient-poor soil in the absence of added nitrogen fertilizers. His research was recently highlighted in Science magazine, and his work has suggested that there are several ways that bacteria can form nodules in the symbiotic relationship, and he has findings that have important agricultural implications. So with that, please welcome Mike Sadowsky.
Thank you. So I'd like to thank the Planning Committee and Jeff for inviting me to speak today. And although the introduction talked about my work on nitrogen fixation, I learned early on that there's an intimate relationship between what happens on the land and what happens in water. And my research migrated to water questions quite a long time ago, where we've been studying water quality and the relationship between water quality parameters and microorganisms and how that impacts recreational activity and health. And that's what I'd like to talk about today. So on a global scale, we all know that one of the greatest causes of pollution in the environment is in fact due to the release of sewage-derived microorganisms, which we generally refer to as fecal bacteria, into waterways. And that these organisms are one of the most often exceeded water quality parameters and standards. And when we think about water quality, we also tend to focus on nutrients, but as a microbiologist, of course, I focus on the microbes. And because they often exceed the standards, we're looking for ways in which we can help clean up those environments that are impacted. So we're very fortunate in that Mother Nature has provided a way that we can cleanse the environment by itself in a way, and that if microorganisms enter a waterway, the water will cleanse itself if the load is small. So think about privies on farms, outhouses around the state, or small septic systems. And that seems to work really well. However, microbes can't cleanse themselves if the loads are large. So think of big spills, where we have combined sewage and drainage water entering a waterway, overflowing a system, causing a large, massive spill. We're also fortunate in that microorganisms and a lot of the fecal bacteria are very susceptible to ultraviolet light, and they're rapidly decimated by that light. But in many instances, however, these microbes make their way into the waterways and cause a lot of problems by exceeding standards that result in the shutting down of beaches or the limitation of recreational activity on certain waterways. So the main problem that we've been addressing is that many of the water miles in rivers, streams, and lakes around the state and the nation, and also this translates to international as well, exceed bacterial levels of fecal bacteria. And I'll talk about which bacteria I'm talking about in a second. And that these fecal bacteria standards are most often exceeded in all the water quality standards, as I said. And the main question and the main problem is that all these organisms are treated the same regardless of where they come from. And the technology that we currently have doesn't say where these microorganisms come from. Now, the main problem, I think, and this is my personal opinion, is due to one of urbanization. So historically, human populations lived relatively dispersed, and they had agriculture and hunting survival for their survival. But by 1940, about 14% of the people became urbanites, and by 2030, it's projected that 60% of the world's population will be urban, which then allows us to take a large amount of sewage and concentrate it in a relatively small area, and then there's problems associated with that, many of which are obvious. There's other problems with urbanization. One is that the conversion of land from natural habitats to urbanize leads to habitat destruction. Also, the removal and depletion of natural resources is a big problem. And lastly, the production and disposal of waste products has been and will continue to be a problem in large urbanized areas. In addition, if that wasn't enough, we know that urbanization alters temperature and precipitation pattern, increases pollution in the air, and more importantly, it alters surface properties and prevents runoff, it leads to runoff of water from surfaces into our waterways. So think of parking lots. So think of a major urban center like something like New York City or San Francisco or LA. There's a lot of cement around that leads to a lot of runoff issues. That would be mitigated if it was in a smaller community, but that's not where things are going. So what can we do? Well, I looked at Greenpeace, and I read a piece that they wrote, and they said they wanted to achieve zero discharge, which might be hard. They want to recover excreta as a resource, which many people are doing now, thinking of converting it even to electricity. We want to avoid the use of water. Well, that's going to be hard, especially in a growing region like ours. Disinfect without chlorine and prohibit industrial wastes. So clearly, some of these are attainable. Others maybe are in the future or will never be attainable. But these will lead, at least, to water sustainability and issues that are associated with water runoff and fecal pollution of waterways. So now I need to talk a little bit about fecal pollution of waterways, and I'll try to keep it not very graphic. But the whole idea of fecal pollution in waterways in the United States was initially revolved around this issue of a fecal indicator bacterium, and several hypotheses, some of which have not been tested. And the first hypothesis is that fecal endobacteria are most likely coming from human sources. So if we detect them in waterways, it indicates the potential presence of a human pathogen. It's just a potential presence. And that human fecal bacteria and the organisms that come with human feces are most likely more harmful to humans than that fecal material from other animals. But many of these hypotheses have not been thoroughly tested. We can't test things on undergrads and prisoners anymore. So we're stuck with really doing correlational analysis using epidemiological tools that allow us then to make some type of correlations between the incidence of disease and someone using a recreational waterway or sitting on a beach. And that has provided the whole framework for the United States Environmental Protection Agency and subsequent states' adoption of fecal monitoring standards. But these bacteria are really cheeky, and I said no pun intended, but I think I do mean a pun intended. And then given enough numbers of these microorganisms, they, like any other living creature, is under tremendous selection pressure to either die or find an alternate host. And microbes, although they're small, they're pretty smart, and they want to live like we do, so they actually find alternate hosts for their survival. And that has led to a lot of problems with this whole hypothesis and the whole framework in which we look at water quality. So these indicator organisms, as I said, imply fecal pollution. And the fecal bacteria that we use as indicators used to be called fecal coliform bacteria, but now we focus on two specific groups of organisms. E. coli, if it's a freshwater system, and Enterococci, if it's a marine system. And in fact, the US EPA would like us all to go to the Enterococcus standard. So these are the primary indicators of fecal pollution. And they were chosen because they're easy to grow, easy to enumerate, and you don't need a lot of technology and brain power to figure out how to measure them. Also, these organisms tend to be prevalent in the environment at much higher numbers than the pathogens themselves. So you can use one method to detect these organisms, whereas you'd need hundreds of methods to detect 100 pathogens. So that's why this indicator method came about. However, these conventional methods don't determine sources of the fecal pollutants, just say they're present. So you can't go back and test the first hypothesis, that they came from animal or human sources. So there's a lot of other problems associated with these classical fecal bacterial tests. First of all, they're time-consuming. They take about 24 hours to get the results. So if I go into a waterway, a beach, a river, or a stream, and I take a sample, I can post the results tomorrow for what happened today. So I'm always a day off. So if the beach says closed, or it says no recreational activity, you actually don't know if that's true or not today. It was true yesterday. There's a lot of false positive reactions. Sometimes it underestimates the degree of contamination. And more importantly, it doesn't identify a pathogen. It just identifies an indicator, right? So you may or may not ever get sick from that waterway. Okay, so this is the main issue. This poop in our soup, where's it coming from, right? And there's a bunch of potential sources you can think of easily, relatively large ones, and most people point the finger at agriculture, but that's just one of them. It could be wildlife, and we know in Minnesota, for example, in this time of year, we have a lot of ducks and geese visiting us. That's a huge source of fecal inputs. We have domesticated pets, and that's a real problem for people who like to flush their animal material down the storm drains and so forth, or don't pick up the poop when their animal makes it. And of course, one of the large ones is humans, and then there's a lot of wildlife that we tend not to look at. So it's mice and voles and all little things that contribute to the total loading, and those lead to excessive amounts of non-point source pollution. So the methodological limitations I just told you about can't distinguish between any of these sources of pollution. So if we're to believe the central hypothesis that you're most likely to get sick if you ingest fecal microorganisms from a human versus another animal, and I don't know if that's true because some animals like birds carry salmonella, then what are we going to do about it? And that's where my laboratory's research is focused on for the last 15 years or so. So really the hope for the present and for the future are some of these new molecular technologies that you've heard about, have the necessary both sensitivity and accuracy to differentiate among these bacteria that just inhabit different GI tracts. So they could be the same organisms, but have slight differences because they either evolved with a human or evolved with an animal, or have some other distinguishing characteristic that lets me say this came from a chicken and this came from a human. Right? And these organisms are essentially the same. So it's the same genus, the same species of organism, but has a slightly different habitat. And can the technologies work to help us? And this is a work in progress. There's not a lot of answers that are definitive yet, but we're getting there. And this whole concept brought up this whole new area of research called microbial source tracking. Now, this has in its infancy studies on epidemiology of foodborne diseases, where you can go in and look at a tomato and say, yes, it has the same organism as all the people that got sick. Right? And we're applying that now on a larger environmental scale. So essentially, microbial source tracking represents a series of approaches to define the origins of non-point fecal contamination. It's easier to deal with point sources. You can see, obviously, that there's a sewage overflow or there's cows standing in the stream. We can use this for microbiological methods, genotypic methods using molecular DNA technologies, and chemical methods can differentiate these things. And what's driving a lot of this is the change in the government's relationship to the contamination, and that now they want us to apportion sources with a TMDL, a total maximum daily load determination. And if you don't know where the sources are and where the fecal bacteria are coming from, you can't deal with the TMDLs, and you can't institute best management practices to fix it. So this is really important if you want to address the problem. So these methods are needed essentially to meet TMDLs and establish load allocations for both point and non-point sources. And TMDL implementation is difficult. And in fact, the regulations preceded the science, and that often happens. So TMDL regulations came about, and then the science followed after in trying to develop the tools to actually meet the TMDLs. And that's the history of this particular area of research. So in microbial source tracking studies, the most important thing you can do is ask the right question. And we developed a series of decision tree type analyses to help guide both local, state, and federal agencies in how to address this. So one is really simple. Is it human source or an animal source? Yes or no? Human versus animal, for each source is a little difficult question. So what are those animal sources? And what's the proportion of those animal sources? Their percentage distribution and contribution. And the question then designates what types of microbial source tracking method you actually need to address the question. It's easy to say human versus non-human. It's much harder to say 23% cow, 14% pigs, and 12% birds. Right? Okay. So I have to get a little bit into the trenches with a little bit of the technology because there's fundamentally two different types of technologies that people use, and I hope I don't bore you. One is called a library-dependent technology, which I'll talk about first, and the second one is library-independent. So a library-dependent technology is something along the lines of this. The question is, what E. coli are in this waterway? So one way to address that is to isolate E. coli from a large number of known animal sources. And we've done that for 17 animal sources. And then we use the same technology that the Bureau of Criminal Apprehension uses, that is DNA fingerprinting, to fingerprint every one of those E. coli strains in the host library. Then go into a waterway, see what organisms are present, what E. coli are there, and then fingerprint them and see if they make a match to the library. So that sounds really easy to do. It's actually not, because of several really important problems. That is, these technologies only work if genetic diversity is limited. That is, there's only, I'll make it up, 20 different kinds of E. coli out there. Then I can pinpoint which one is which. Also, it's based upon the hypothesis that for E. coli, there's host-source-specific organisms. That the E. coli in your gut is different than the E. coli in a chicken or a bird's gut. Okay? And if the first one is true, then at some point the library is going to reach saturation. And what I mean by that is, if I go pick another isolate, I'm already going to find it in my library. So I can stop picking isolates. Also, the library has to capture all sources. And for this to work, the majority of what we consider now unknown becomes known. And we can apportion a source to that. But despite all we've done, library-dependent methods generally fail because of several major issues. Right? So these new tools that we have become much more limited. They can't address all those questions. And the reason why we can't address those questions using a library-dependent method for E. coli is that simply there's many, many sources of E. coli that are present in the environment that have nothing to do with an animal. For example, some studies we did up in Duluth Superior Harbor, funded by Sea Grant, showed that in one stretch of the waterway, 40% of the E. coli that were there were coming from soil. So despite what you might have heard or learned in microbiology classes, E. coli and enterococci are not limited to the intestinal tract of warm-blooded animals. Therefore, the question is, should they even be indicated to water quality? So we've shown through various research, based upon initial studies done by Roger Fujioka and others in tropical environments like Hawaii, Guam, and studies done in Florida, that temperate soils are sources of E. coli. Even in Minnesota, I can find E. coli living in soil. They freeze over the winter. When they thaw in the spring, the numbers start to grow again, and those, as I say, could introduce up to 40% of the microbes back into the waterway. So if I just count E. coli, I'm not counting where it came from. I don't know which source. For those who have visited Lake Michigan and the other Great Lakes, you see this stuff growing up on the shore. This is the macrophyte alga Cladophora, and it can hold anywhere up to 10 to the 9th E. coli per gram of Cladophora, a huge source, and also human pathogens. And I hope you don't see these signs here where the wind is nice, the temperature is pretty good, and the sewage is moderate when you visit those beaches. But for those of us who study beaches, and someone really has to do it, sand and sediment acts as sources of E. coli all around the beaches around the world. So we actually did a couple of studies around the world on both the bacteria and the fungi that are present on the beaches, and it's not pretty. Okay. And in Lake Superior, where the water is too cold to have that macrophyte algae, you'll find this, I call it angel hair type alga growing on rocks. It's really periphyton. It's a mixture of algae. It's a mixture of diatoms and bacteria and slime. This is stuff that makes you fall on your butt when you get into the waterway. It carries a lot of E. coli as well. So are there other sources? So originally we're taught that they come from warm-blooded animals. How about cold-blooded animals? Well, I had a student I gave a fishing rod to and a fishing license and said, go catch some fish. And he found E. coli present in a bunch of different fish species, both benthic and pelagic. Right? And of course, birds are a major source of E. coli. And we follow bird migration across the Great Lakes and can actually track their E. coli and other organisms as they move around. So there's many, many sources of E. coli in the environment. So again, using them as a fecal indicator might not be a good idea. So I really think it's safe to say that E. coli is everywhere, right? And Enterococcus is even worse because it can survive in most environments that E. coli can never survive in. That is, desiccating environments, low nutrient conditions, and that's why it makes such a problem when it gets in your body and causes diseases in which antibiotics don't work anymore. So you might have heard of vancomycin-resistant enterococci, VRE. This is becoming a real problem now. So we know that there's limitations to these library-dependent methods if you focus on a single organism, and that the library is not large enough to capture all the stuff that's shared between different animal hosts and those we call cosmopolitan organisms. We know that the library is not representative. The libraries need to be regional, and sometimes they're directed at the wrong organism. For example, if you're in a warm water environment and there's E. coli growing on the soil, why are we looking at E. coli? Maybe we should look at something else. Okay? So these limitations to library-dependent approaches makes us take some independent approaches. And what we've developed over the years are a series of molecular markers that indicate the presence of bacteria that are not E. coli, but actually represent the organisms that are the most prevalent in your body. So I always feel like Carl Sagan when I say this. Remember, he looked up at the sky and said there's millions and billions and trillions of stars. Well, there's actually more bacteria on the Earth than there are stars in the universe. And of those bacteria, one of the greatest ones belongs to a group of organisms that inhabit your intestinal tract that are approximately present in 10 to the 9 to 10 to the 12 bacteria per gram of fecal material. That's an amazing large number. I can't even imagine what 10 to the 12 looks like. Okay? But those bacteria are specific for the host, and we can track those in the environment. And that we use a technique called the polymerase chain reaction, which is becoming a standard method of practice in places like California. It's becoming practical now in Wisconsin, and soon it will be moving over to Minnesota, I'm sure. So the analyses that we did before, dependent upon the growth of E. coli, limits what you can see. So other organisms really need to be looked at. And the fundamental question we asked in my lab was, why look at one organism? Let's look at all the organisms that are present in an animal species and in a waterway and try to make some type of relationship between those two parameters. And can we do it? So fortunately, a new technology was evolved several years ago, and that technology is referred to as metagenomics. So I'll make it bigger here. So what is metagenomics? Well, really, it's just the study of the totality of the DNA and the fragments of DNA that are present in any given environment. So there's metagenomes on the table you're sitting at. There's metagenomes on your earlobes, on keyboards. Everything has a metagenome. It's defined operationally by the space you're looking at. And there's different types of metagenome analyses. we can focus on questions of diversity of microorganisms by looking at one piece of DNA that taxonomically defines all microorganisms. It's called the 16S ribosomal DNA. We can study all the microbes together as a community, or we can look at specific genes that carry out specific functions, like genes involved in nutrient cycling. Right? So why use genetic metagenomic analyses? Well, I gave you some preamble to that a second ago, but one of the most important things is that despite the fact that we've been growing microorganisms in pure culture since the time of Louis Pasteur, and he was looking for diseases of wine, we can grow much, much, much less than 1% of all of them. And that's due to our ignorance on what those organisms require to grow them. So these cultivation-dependent methods that we've all now kind of grown up with and used for water quality standards throughout the world really influence what we're looking at, because it's only looking at a narrow number of organisms. And more importantly, if you look at all the organisms in the intestinal tract of most warm-blooded and cold-blooded organisms, animals, you'll find that E. coli represents only a tiny bit of what's present. And in fact, many times you don't even see it. Right? So we focused on E. coli because it was easy to grow, but I think we made a major mistake. So the approach that we take for these types of analyses, excuse me, in essence are quite simple. You go to the environment, you extract the DNA, you sequence the DNA directly, or you make various libraries of the DNA and ultimately sequence it. And what you're left with is a large number of sequences that you have to sort through. So this is typically called high-throughput, large-scale DNA sequencing technology, which over the years has become very, very inexpensive. It's not quite at the Star Trek level yet, where you put a sample in and 2 seconds later the answer comes out, but it's pretty close. It's just within hours right now. Okay? And they make these technologies so cheap now that for $1,000 you can buy a DNA sequencer that plugs into the USB port of your computer, right? Which is quite astounding. So the different type of technologies vary. What we use is a Lumina-based sequencing technology. That's a company. I don't work for them. All right? That's my disclaimer right there. The platforms that we typically use is called a HiSeq platform, which will allow us to analyze 200 samples simultaneously. There's other more rapid technologies, one called the MySeq platform. It sits on your desk. It's slightly bigger than a laptop and a screen. And it gives you results in about 24 hours. And then the new technology that's come out is called the NovaSeq. And this number is hard to get a grasp on. But every time you run it, you get 1.6 billion DNA sequences. So this is really big data, and to analyze this big data, you need a whole new series of tools and a whole series of statistics that you probably never wanted to learn, but you have to use them because you can't put everything down on one piece of paper if you got 1.6 billion or anything. Right? So I want to kind of go through that technology right now, just kind of covered a little bit of how you deal with hundreds of millions of billions of DNA sequences and how you can make some sense out of it. I have to put up this tree of life, and this is a tree of life that represents both the bacteria, their primitive cousins, the archaea, and the eukaryotes, where we humans stay up there on the top up here. And I'm only going to present bacteria only in this talk, and I'm not even going to talk about the other domains of life, which are huge as well. But as far as genetic diversity goes, bacteria takes it all, because there's so many of them, and they're so diverse. Right? So the overall goals of this technology harken back to this library-dependent technology I told you about. But the libraries now are not going to be made of E. coli bacteria that I pick out. And by the way, you need about 10,000 E. coli strains to saturate diversity. And I don't know about you, but my students would never pick 10,000 of anything if I asked them. Right? They'd all run away and go work with Jeff or something. Okay? So the goals in this project is not to pick out individual E. coli, but to look at all the DNA sequences in their totality and then try to match up what's present in the waterway with what's present in animals. Right? So to do this, I have to introduce this concept to you, that there's DNA sequences out there that I might not know where they came from. And I just define them operationally as a potential taxonomic unit. I don't know what it codes for or what organism it came from. I know that I can stack up a lot of them together, and they're called shared OTUs. Okay? And I'll show you that in this picture here. So this is a network analysis of where I took OTUs, operational taxonomic units, that came over DNA sequencing of a bunch of farm animals in Korea. And then I looked at which OTUs are shared between the farm animals and which ones are shared with the waterway, right? So this type of analysis is quite easy to do now. It's just putting a button on a computer. But this whole idea of going through large number of data sets required the evolution of a new series of tools, which we generally put on the name of computational biological tools. So you have to learn how to use these tools. And one of these programs came out, CD-HIT, which allowed us to look at all these OTUs and rapidly determine which ones overlap. When we tried this the first time, it took us days and days and days to get analysis. Actually, the first time I tried this, I killed one of the Minnesota supercomputers. Right? So these new programs speed up the technology a lot and allowed us to develop tools using DNA sequencing and shared OTUs to determine sources of bacteria in watersheds. And this first experiment I did in Korea with some of my former students, and we were able to look at urban areas, open areas, and agricultural areas, and got pretty good matchup between what the program told us, what the major source of impacts for humans, wildlife, in this case geese, and in the agricultural area, the major impact was pigs. So that started us down this whole line of trying to refine this technology and use it here in Minnesota to address some of the major problems that we have with fecal contamination. And then one thing we wanted to do also is to provide a resource for our colleagues. We wanted to develop these libraries, put them up on databases, and then have someone just go into a waterway, have all the DNA sequenced, and it is really inexpensive. You could do it for $80. And then throw it up against this library system that we have. So we have an automated microbial source tracking system. And then people can get the answer spit back and say, your most likely input source is X animal. Okay? So this pyro-MST method was our first attempt to deal with this. But as we were doing this, the technology changed. And that is that the method of DNA sequencing, which used to be called pyro-DNA sequencing, changed. And the number of sequences expanded exponentially. So you've all heard of Moore's law, right? Which tells you the relationship between computational power and cost. Well, DNA sequencing technology is advancing faster than Moore's law. So more rapid technology advancements are generating even cheaper and cheaper technologies. So as the technology changed, we needed to develop new bioinformatics tools and methods to look at this. And the first place we applied this, for an example, is the Minnesota section of the Mississippi River. And due to some nice funding we got from the federal government, we actually got some of that shovel-ready Obama dollars, right? And funding from the LCCMR, we started the Mississippi Metagenome Project. And if you go across the street to the Science Museum of Minnesota, we actually have an exhibit there for that as well. And the whole idea was quite easy to do because we wanted to see the impact of human activity on the microbiota present in the Mississippi River, both fecal microorganisms and non-fecal microorganisms. And this technology allowed us to do that. So the headwaters of the Mississippi are relatively pristine, but we know that as the Mississippi River passes down through the U.S. heartland, anthropogenic activity alters both the structure and the function of those microorganisms present. And we wanted to have an understanding of that. And prior to these sequencing technologies, we could not do that. So we know that the confluence of all the rivers into the Mississippi are likely to have a major impact. And we focused our attention on the Mississippi River within Minnesota boundaries and the confluences of the rivers within the Mississippi within Minnesota. And we know that Minnesota has an impact, too, in the Gulf of Mexico and is proposed to be one of the major sources of nitrogen loading to these waterways, ending up in the Gulf of Mexico, causing these dead zones. So again, we focused in this case just on the bacteria. That was enough. But if you want to go back and look at the archaea and the eukaryota, we can. And we're providing these libraries to anyone who wants them. So the M3P project, I couldn't use 3M for obvious reasons, allows us to ask how the activity alters microbe structure and function, nutrient cycling, persistence and fate of chemicals in the environment, antibiotics, human hormone mimics, and more importantly, questions relating to the whole ecosystem functioning. And that's the kind of stuff you can get out of just doing DNA sequencing these days. So I'm sorry the map looks a little bleached out, but essentially we started up here in Itasca and made our way down to the bottom of the state when the Mississippi leaves the Minnesota. And the first question we had was, how many sequences do we need from each water sample in order for us to saturate genetic diversity? So I told you E. coli and other organisms are very diverse. And the Mississippi River is actually second in diversity to soil and contains about 5,700 species of microorganisms in every 100 milliliters of water. Quite astounding. Right? And if you get 200,000 sequences from each sample, you can pretty much saturate genetic diversity, as you see in this curve, becoming asymptotic. Okay. And if you just look at the Mississippi River up from Itasca versus the Twin Cities, you can see that they're vastly different. And they share some things in common, which makes sense because the river's flowing down into the Twin Cities. So you have to add up Itasca microbes to every microbe that comes after that. And you can end up with bar charts that allow you to get some idea of what classification of bacteria are present at each site, which bacteria might be unique to a specific site, and maybe that can give you insight into which microbes are loading the waterways at that site. And then you can relate all this data to any metadata that you might have. So in this case, we did it to land use. Right? So you can see the relationship between the individual microbes and land use. And what's really nice about this technology is that you can check it a little bit and see that the urban sites tend to group together, the northern agricultural sites group together, and the southern agricultural sites group together. And this principal component analysis is the only way that we can look at millions and millions and billions of bits of data, where everything's summarized to one dot. So this one square that says St. Cloud, sorry, kind of not getting bright enough here, represents all the microorganisms present in the waterway at the St. Cloud station site that we looked at. Okay? So it's a summary. And more importantly, using some new technologies based upon network analyses and what's called Bayesian inferences, you can then assign individual factors to each microorganism. I can say this E. coli strain or this Salmonella strain is more influenced by the calcium level in this water than anything else. And then you can start looking at what's driving the changes. Right? So understanding metagenomics is of primary importance. And using this technology, similar to what we did in the Mississippi River, we decided to use this to make what we call next-generation fecal taxon libraries. It is a series of DNA sequences, hundreds of thousands to millions, that come from multiple animal hosts. And then the idea then is to make a library of these taxa and then see what's in the waterway. And I told you that some of these taxa are unknown, so we call them operational taxonomic units. And it gives us information both about fecal microorganisms and environmental bacteria. It doesn't separate the two. It just looks at bacteria. And we now have been addressing to use this technology in microbial source tracking. And I think we have some success. So the research questions are simple. We want to ask essentially what's causing these polluted waterways and what's the animal inputs. Are bacterial communities and environmental bacteria similar or are they distinct? If they are distinct, can we differentiate the animal sources like we said before? And can the fecal microbiomes of different animals differentiate one group of animals from another? That's our basic question. Right? So again, our studies were done up in the Duluth Superior Harbor area. What I presented before is very similar to this. You go ahead and get a DNA sample, you sequence the sample after isolating the DNA, and then you do the data analysis. So I'm making it sound really simple, but I have a group of people in my lab that do nothing but, pretty much so, bioinformatics to address this. And I have to thank the Minnesota Supercomputing Institute for allowing me to have supercomputers to do this, because they do take a lot of computational power. Okay. And ultimately, in the end, we want to apportion the amount of material coming from dogs, human, and gulls, for example. Okay. So we went to several sites up in the Duluth Superior watershed, including those in the St. Louis River estuary, those surrounding the sewage treatment plant in the waterway, and there's two. There's the Western Lakes Superior Sanitary District dealing with Duluth, and then there's the Superior Sanitary District dealing with Superior, Wisconsin. We went to sites that were typically polluted or considered contaminated. One is the Southwood Marsh site, one is Minnesota Point, and then there's some Storm Creek drains that have been shown to have a lot of E. coli input. So we wanted to examine them as well. Okay, so Southwood Marsh has a storm drain. It's typically reported to have a large amount of pollution around it. And sitting around that storm drain, of course, is a lot of wildlife. So we're talking about ducks and geese surrounding there. The Brewery Creek storm drain also has a lot of E. coli in the waterway. We think that they're actually propagating within the system itself. So we wanted to look at that as well. And I'm not going to go through all of these, but we also looked at the harbor and also looked at Lake Superior itself to see what's the impact of microbes, what microbes we can find, and can we use this new type of microbial source tracking to deal with this. So I don't know how well you can see this, but this again is a principal component analysis that is relating different groups of organisms to different sites. So these are the DNA sequences we came up with from WLSD, and then we have those that are characterized by organisms from different animal sources, and we can group them together and get a pretty good idea of what's the input sources to these waterways. So we can look at the same type of bar charts that we showed before, and the one on the right here is the WLSD pumping station, the effluent station, and these are the key signatures of microorganisms. And each one of these bars represents a different group of microorganisms present. So you can look at these bars and try to make a judgment, but we do it computationally. And in the end, we all resolve it back to these principal component analysis that shows that we can actually look at these E. coli and other organisms present and say whether they're environmental sources, whether they're fecal sources, or are they shared between the two? Right? And that allows us then to pinpoint where these organisms come from. And if we have a library, we can say, okay, some of these came from birds, dogs, deer, and some of these came from other animals. And that's where the technology really exceeds. And this is a cluster analysis of the DNA sequences in our library that came from geese, chickens, effluent, beavers. They're really fun to work with. And a lot of other animals. Some are very clearly distinct and others are mixed together. So if we focus on the distinct ones, we can get definite good data out of this. Right? We also noticed that we can look at some animals and see some variation, although beavers are pretty close together if you just look at the colored bars and the relative percentages, while other animals are less so. And there's wide differences in the microbiota that are present, for example, in turkeys, than those present in swine, okay, or sheep, right? And some of these animals have unique taxonomic units that allow us to focus specifically on the microbes that come from them, right? And raccoons are really diverse, most likely because they eat anything. So their gut microbiota reflects what they eat. So you are what you eat. That's definitely for sure. Okay. So let's move on now and show you the results of our studies up at the Western Lake Sanitary District and Duluth Superior Harbor. And if you look now, we did multiple runs of the Source Tracker program. So now instead of using this PyroMist program, we use a program that was developed by one of our junior faculty members here named Dan Knights, who's been working on this whole metagenomic area for a long time. And he developed a program called Source Tracker, which we now use all the time. And running Source Tracker over and over again, we can get a good idea of what input sources are impacting different sinks. Okay? So here's the listing of the sites on the top. The blue bars, unfortunately, represent signatures of wastewater effluent. The green bars in this case represent signatures, these dark green bars, of gulls, and the light green bars represent geese. So you can clearly see what's impacting our test sites at this particular area. I'm not making any comment about whether there's pathogens or not. I'm just trying to apportion sources to these particular animals. Now, WLSSD actually does a good job at cleaning up wastewater, and it removes 99.99% of the bacteria present in the influent sewage and the pathogens, but it still dumps a lot of microorganisms in the environment. All sewage treatment plants do that. And you can also look at it in other ways. For example, you can look at these type of trees, and on the lower right, again, this is not very bright here, you can see WLSSD clusters with a lot of the other sites. So it's a major impactor on the microbiota present on these sites. Okay. The other thing that we wanted to do was to do power calculations. If you want to make a fecal taxon library, how many animals do you need in each library to adequately capture the diversity of organisms present? And fortunately, when you do a power calculation, you find out it's around 13. So with the E. coli libraries I talked about, you need 10,000 E. coli strains. Now I can go and just get 13 different animals, types of each species, analyze those, pull them together, and that's my library. So it makes it much more economical and easier to do. All right. And lastly, we had just published this recently in Environmental Science and Technology, so if you want to read more about that, this is our first paper in the United States. We've done several studies in Australia, several studies in Korea. And this is an international project, and I just finished a beach study in Portugal. Okay. We also know that the number of taxa that are present in a waterway is directly related to what influent source there was. And this tells us that rather than looking at one organism like E. coli or two organisms like enterococci, the source tracker program looks at hundreds of markers to come up with its answer. So it's much more adequate way to look at things. We're not saying it's only E. coli. We're saying of these 134 organisms that make up what we call effluent from this one site, we can detect those in waterways. So it's much more accurate. So in conclusion, we can say that bacteria and animal fecal samples and environmental samples are different, but some cluster together. And that clustering allows us to partition sources, and that subsequently allows us to deal with TMDLs and BMPs. We know that the community structure in all these animals is different. The human is unique, but it's shared with your companion animals. So it's often difficult for me to distinguish the microbiota in a cat and a dog in your house than your own, and I won't go into how that happens. But your pets lick you, they share boxes in the house, they share your food, and you're much more intimate with them than you are with cows or geese. Right? And that the differences in abundances of organisms that are less abundant are useful to differentiating. So you don't look at what's the major organism present in that species. You look at all of them, including the ones that are only in a small amount. And that way you get a better picture of the relationships. So we do know from these studies that if you want to look at fecal contamination, we know that diversity is huge amongst all these organisms. But some diversity is narrow, especially amongst the organisms that co-evolved with humans, right? Or sheep, or dogs, or goats. But also keep in mind that the bacteria were here hundreds of millions of years before animals were. So their evolution occurred independent of the animal host and then subsequently dependent upon the animal host. We know that bacteria are everywhere. In fact, that's one of the hypotheses, the universal bacteria, everything is everywhere concept. Right? And will this impact water quality and wastewater microbiology? Absolutely yes. And some of these new tools is our way to see what's really going on without looking at one or two microorganisms. And does it matter for public safety? The answer is absolutely yes. And new technologies that look at individual pathogens are now available. They're not rapid, they're not cheap, but we can go into a waterway and say exactly what pathogens are present and couple their presence to the relative numbers that we find using these metagenomic technologies. So we're going to get better at refining safety issues. So these next generation fecal taxon libraries that I talked about contain all the taxonomic units of pooled fecal samples from known animal sources. And our recent studies said they're sub-regional. In other words, if I make a fecal taxon library for Duluth Superior Harbor, it might be different than that what's present that I have to make in southeastern Minnesota, because the organisms are eating different things and the environment's slightly different. But since you only need 13 animals from each one, it's pretty easy to make that. Okay? And if you can run 200 samples at one time, adding 13 of each individual host is nothing. This gives us information, though, about all the potential microorganisms in the fecal and the environmental sequences. And these samples are fast, cheap, and very accurate. And this is going to lead a whole new way as the cost breaks down. And we make this more simple. And I call it PhD technology. That is press here dummy, right? Where you just put the data in, you hit a button, and out comes the answer for you, without having to worry about the statistics that you really never wanted to learn about. But I just wanted to point back to you, despite this fancy technology, the best technology was provided by God to all of us, these two peepers right here. And you need to go out in the environment and look at it and figure out what are the potential input sources without doing all the DNA stuff first. And this poor guy left his car, went on vacation probably, and it came back saturated with pigeon material, and I didn't need any fancy technology to figure that out. So with that, I'd like to acknowledge the people that allowed this to happen. So I just sat in my office and wrote papers. It was really done by Clarissa Brown and Chanlan Chun, and the whole Mississippi Metagenome Project was done by all these individuals, and that was a multi-year project. We have received funding from the College of Biological Sciences, from CFANS. We got federal stimulus funds to do one of the few projects that was federal stimulus fund money, and I was very proud of that. And we got funding from the LCCMR and also nice funding from Sea Grant. So I'd like to thank all these folks for providing funds for allowing us to do our research. So with that, again, I'd like to thank the organizers for inviting me to speak today. I'd like to thank you all for showing up and not falling asleep. And I'm happy to answer any questions. So thank you very much.
All right. Thank you, Mike. Great talk. And we're going to turn our attention to Pigeonhole here, where we've got some questions for you. Are you really seeing illnesses of concern due to microbial contamination?
I personally have not been seeing those, but those doing epidemiological studies have. So I forgot to mention that the majority of these regulations came about due to epidemiological studies done in the 70s on the east coast of the US. More recently, they started another epidemiological study in Lake Michigan, and that got halfway or three-quarters of the way through and ran out of money and stopped. But there's definite correlations between certain fecal microbiota and illnesses. Again, they're statistical correlations. They're epidemiological studies. But there are illnesses, and that leads to beach closures.
Thank you, Mike. We'll go to the next question here. Quantitative data are what managers need. Genomics appears to only provide relative data on sources which doesn't inform on actual risk. When will we be able to quantify amounts of harmful microbes?
Okay, so I just briefly got to that at the end of my talk. And there are technologies now that can actually measure the number of potentially harmful microbes in the water. But you have to remember that there's one major problem. And that problem is those harmful microbes in the waterways are present at very, very low numbers. So it's difficult to detect unless you have some technology that can concentrate those microorganisms. And that's what people are working on now. What looks very promising is a technology that came out of the kidney dialysis area, and that's used something called tangential flow, not centrifugation, but tangential flow filtration. So essentially, you can run hundreds of liters of water through a dialysis column, which is made up of a million little tubes, concentrate the microorganisms, and then subject that to quantitative PCR in these micro platforms, nanotechnology platforms. And you can tell exactly how many Salmonella, Shigella, and all those organisms are present. It's still at the testing stage, though. It's not ready for prime time, but soon.
Thanks, Mike. Do you have an idea for a standard to replace E. coli for swimming and drinking?
This has been a tough question. So we've involved the EPA in trying to help us make these decisions. And the EPA said that you can go off the E. coli or Enterococcus standard if you can provide another indicator that would do better than those in really relating health potential risks to the microorganisms. So the only ones that have tried that so far are Hawaii, where E. coli and Enterococci are everywhere, and they just started to use the organism that you might have heard of. It's called Clostridium perfringens. It causes foodborne outbreaks, but it also is present in the environment. And they're trying to move that through. But right now, we're stuck with the E. coli and Enterococcus standard until people start to realize that other technologies might be better. And that's going to be driven by cost, ease of use, and availability.
Makes sense. It seems that MST techniques have still not developed to the point of practical application in most routine water monitoring and management efforts. What would you suggest we do in the meantime?
Great question. So there are microbial source tracking technologies that are not based upon the DNA technologies I showed you about, but other ones based upon quantitative polymerase chain reaction, PCR, you've all heard of that, right? Those technologies work, but they're expensive. So I tend to tell people that they should couple a few of those along with traditional monitoring to get an idea of what the potential input sources are, and then focus your TMDLs on that. So until the prices of those technologies drop down so they're cheap, easy to get, and rapid to do, we're going to be stuck with using a few of those samples, and those are done by several labs within the state and outside the state, along with traditional coliform monitoring. We're just not quite there yet, is what I have to say. Okay.
So, Mike, you have pinpointed where specific loadings are coming from. Now, most importantly, what is the next step? Eradication of the Canadian goose or raccoon populations?
Well, eradication is a relatively severe word. So some people have developed strategies to remove Canadian geese from the site where they're making the major impact. So for those of you who live up in Duluth Superior area, think of Duluth Boat Club. That is inundated by geese every year. And there's some simple things you can do. There are dogs that can run up and down the beach, and they love to chase birds. Birds also hate monofilament line, fishing line. And if you string up some of that above head height, you can deter birds from coming. Some people are advocating the uses of fireworks. Raccoons is another issue. I have no idea how to get rid of a raccoon. Right? Those organisms live with us. I think we need to get the questions answered whether organisms present in a raccoon have any impact on us at all. And then you can start targeting what you want to try to move away from your sites of impact. We are not getting rid of non-point source contamination because it's due to everything. Let's focus on the point sources and things we can do things about, like feedlots, human sewage overflow, leaking septic systems. Those we can target.
Sounds good. I'm hoping you weren't the one that was following the beavers around, were you?
No, we actually worked with DNR. Okay. So for many of the animals, I should say, we went to the state fair because that has the largest collection of animals throughout the state. The other ones, we partnered with both Minnesota and Wisconsin DNR, and every animal we have in our library is trapped or collected live. We don't collect anything on the ground. So we have really fresh material. And beaver poop, by the way, is about 99.9% wood chips, as you might imagine.
All right. Thank you, Mike. So the next question up here is a little bit related to the question before about the E. coli standard. Are you working with MPCA to redefine the impairment standard and sampling protocols?
I'm not trying to redefine their standard. I'm trying to help the MPCA in applying the guidelines in a proper manner. In other words, if E. coli counts say that this whole watershed's impacted, I'm trying to work with them to point out where the E. coli counts are coming from, using some of these source tracking technologies and other organisms present, and say, maybe this shouldn't be listed because there's no animals that we can do anything about in this watershed. And we've done that a couple of cases now. So MPCA has been really great about working with us and trying to adjust the regulations to where they needed to be used properly.
Okay, thank you, Mike. This will be our last question here. It has to do with MCES treatment plants and their chlorinated effluent before discharge. Can you distinguish between dead and living organisms, or are the dead organisms available as an indicator?
So in our DNA sequencing technologies, we don't differentiate between a live and a dead. But in our quantitative PCR technologies, we do. We also know very clearly what the decay rate is of each of the indicator organisms or each of the indicator animal species. And one nice thing about the DNA is it doesn't last very long in the environment. So it really gives you an idea of freshly released material. Right? But we can differentiate live and dead in some cases.
Great. Thank you very much, Mike. Join me in thanking Mike for his great talk this morning.
Thank you.